monthly webinar series from the micro nano biofluidics group. Today we have with us Professor Dino DI CARLO from University of California, Los Angeles. DI CARLO received his PhD in bioengineering from the University of California, Berkeley, and then he conducted postdoctoral studies at Harvard Medical School. And at present, he is professor and vice chair in the Department of Bioengineering at UCLA. And he also vice chair in the same university, UCLA. He's a pioneer in the use of inertial fluid dynamic effects for the control and separation of cells in microfluidic devices. His research extends many fields, including lab on particle technologies, directed evolution of cells, rapid diagnostics, and so on. He's a co-founder of several companies like Cytoval, Vortex Biosciences, and so on. Professor DI CARLO has won several awards: National Science Foundation Faculty Early Career Development Award, National Institutes of Health Directors New Innovator Award, Coulter Translation Research Award, are a few among them. He is also a fellow of the American Institute of Medical and Biological Engineering, and is also a fellow of the Royal Society of Chemistry. So let's welcome Professor DI CARLO for his talk on lab on a particle technology to scale biological research thousandfold. Professor DI CARLO.
Thank you so much, Professor Ranganathan, for the really kind introduction and for the invitation to speak. And I apologize for maybe the early hour, but it's very difficult to find a time that works for both of us. So I'm glad that this could work out. Yeah, so it's really a pleasure.
This time just suits us, just starting today start for most of us. So it's fine for us. And instantly it's heavily raining here. So there may be some internet issues, but we'll work with that.
Okay, well, yeah, let me know if there's...
We are recording. Is it okay for you? This video is...
Oh, that's fine. Yeah. As long as it's not, posted publicly, I think it's fine. Yeah.
Okay. Yeah.
Please. Great. Yeah. So it is really a pleasure to come and speak to you today, and hopefully you'll have a nice week ahead of you. So I guess one thing I want to start off with is the work that I have done in the past has been in this microfluidics field, and one of the kind of the key aspects of microfluidics is this ability to make small volumes of fluid, and that's been one of the areas that has led to many applications. So whether it's single cell analysis, whether it's analyzing individual molecules, these digital assays, digital PCR, digital ELISA. So over the last 5 years or so, we started in our lab to think about how we can do microfluidics without microfluidics. And so this idea of using particles as an approach to compartmentalize and create these small volumes, that's kind of where this impetus and this idea of lab on a particle came from. And in some ways, I came from the microfluidics community, and we've been doing a lot of microfluidics, but I've learned over my time in commercializing microfluidic devices that even though you might start with a small chip, what usually builds around that small chip is a big instrument that is very specific then for one application. And so that leads to a lot of barriers for adoption of these technologies because of the cost of these big instruments. And so one of the other reasons we wanted to do microfluidics this way is, can we get around this challenge of a highly expensive, complex instrument that is going to control the assays that are only very specific for one type of test? And so this is, this is kind of maybe the groundwork for what led us down this direction to explore lab nanoparticles. So when we think about where we want to have impact with this type of technologies, I like to show kind of this general slide of what I refer to as the 3 pillars of medicine. And we have molecular therapies, which is what the pharmaceutical industry has been doing for the last decades, making small molecules, making RNA interference or mRNA as we see now, or making recombinant proteins, in particular monoclonal antibodies. In the US, it turns out that now 5 out of 10 of the top grossing drugs, the drugs that make the most money, are based on monoclonal antibodies. And so it's really important if we think about how we can have impact by using the technologies like that automate life science research, like microfluidic technologies. Well, maybe if we can have an impact in the development of monoclonal antibodies, that can be really, really helpful. The second pillar is gene therapies. If you think about this, the central dogma, molecular therapies have an impact maybe at the protein level, potentially at the mRNA level. Gene therapies have impact further upstream at the genetic level. We're seeing huge advances in genome engineering technologies, including CRISPR-Cas9 and many derivatives of this, and new approaches to deliver viral approaches that have been more successful in the clinic, even leading to the first FDA-approved gene therapy by Spark Therapeutics in an ocular disease. So this is an area where we see a lot of potential and where automation tools and microfluidic tools could be very important. In particular, you can imagine, it turns out there's actually a big barrier now in getting virus for gene therapies that is under good manufacturing practices. So the production of virus by cells that secrete them is a barrier so manufacturing ends up being a barrier for the development of new gene therapies and bringing them to the clinic. Cell therapies is this third pillar of medicine that we're seeing huge advances over the last really several years, where we have a lot of therapies that are based on chimeric antigen receptor modified T cells, or we call CAR T cell therapies, where we have a recognition element on the surface of a T cell that then triggers its cell killing function or other functions that are activated by the T cell receptor. So these types of therapies have been really successful in hematological malignancies and treating a number of different types of hematological malignancies. And the exciting part is that cells, unlike molecules, they respond to their environment. They take inputs from the environment, and then they can do some type of computation, and then they can lead to an output, whether that's releasing molecules that could kill a cell, perforin or granzyme, or releasing other cytokines that draw in other immune cells to a site. So that's certainly exciting opportunities. We can also have cells that are engineered to secrete other types of therapeutic molecules, growth factors, extracellular vesicles, cytokines, enzymes, etc. So cells are in essence living drugs, in that they can also expand in the body. So there's huge opportunities as we think about these 3 pillars of medicine and how microfluidics, how using small scale operations on fluid volumes and automating them can have an impact. In particular, one of the biggest challenges for automating our experiments with let's say cell therapies or identifying new monoclonal antibodies is the scale of which we do the biology. So on the right side here, you see the vessels in which we normally do biology, bioreactors, petri dishes, and at the smallest scale, the 1536 well plates. However, the volumes and the length scales at which these operations are done are orders of magnitude higher than the volumes and length scales of biology of single cells. And so that leads to a challenge. That leads to volumes that are too large to really analyze what's happening with the single cell. So what a single cell is secreting, whether it be antibodies or growth factors. They get diluted out into that large volume and are not easily detected. And second, as you have these larger volumes, you also create fewer compartments in which you can scale your experiment. So the scale becomes less as well. And so microfluidic technologies, whether it's droplet microfluidics, microwells, have tried to address some of these challenges where you can make droplets similar to the scale of a cell, accumulate things that are secreted or released from those cells to high enough concentrations to easily detect them. However, some of the advantages of well plates, like having a solid phase that you can capture things that are secreted and perform reactions on, are missing from droplet microfluidics. And so this is an area where we think that the lab on a particle technology can have an impact. And here you're seeing these hydrogel particles, and I'll talk more about these in my talk, but they have the scale of volumes of the microfluidic systems, but also include a solid phase similar to a microwell plate. And so you can now think about analyzing individual cells, but also being able to capture things from those cells, similar to microwell plates. A good way to think about this, so with a 1536 well plate, this is a volume of about 1 to 10 microliters. Our dropicles are these particles that have fluid surrounding them. These are 1,000-fold smaller. They hold about a nanoliter of volume, so enough that a single cell, you can concentrate the signal from a single cell 1,000-fold. And that also, because they're so small, we can make thousands of more of these compartments. And these compartments, these particles themselves, are compatible with instruments that are already in labs. So things like flow cytometers or fluorescence activated cell sorters that allow you to then analyze these compartments and sort them using equipment that are already present in your lab. So this is the excitement that we have for lab on a particle technology. Concentrating signals 1,000-fold by reducing the volume so that we can look at individual cells. Really, the quantum limits of biology, you don't want to look at half of a cell. A single cell is really the limit. And the scale of the biological experiments can increase 1,000-fold as well. So what is a lab on a particle? We think about it as these hydrogel particles. One format that we like to use is these bowl-shaped particles. Here's a 3D printed version of one of these shapes of a hydrogel particle that we make. Obviously, this is not to scale, but you can see this in 3D here. So it has this cavity, and the cavity enables a cell to enter into the particle. These cells can then secrete. We can emulsify these particles. We can put them into an oil phase. Because of the surface energy minimization, they form droplets around the particles that are spherical, and that hold a very uniform volume so that we can further enhance the enrichment and concentration of things that are secreted if we want to, and prevent crosstalk. We can then break these emulsions and do other assays. But this is the general format for lab on a particle technologies. We have some type of particle. That particle then either is interacting with a small volume in a cell or molecules to perform some type of assay. So there's 3 kind of general formats that we've been working on in the lab. Technology we call pico shells, which is an entirely encapsulated particle but is empty inside. Here you're seeing some algae growing within these shell particles. Nanovials, which are these bowl-shaped particles that we can seed cells into without any microfluidic. The end user doesn't need any microfluidics. All the microfluidics is upfront. And then another technology that allows us to barcode and shape particles that have a hydrophilic inner region and a hydrophobic outer region, so we can accumulate droplets and perform assays, but also barcode those assays by the shape of the particles themselves. So this is using another type of fabrication process. Today, I'm going to talk mostly about the Nanovial platform and some of our assay formats with this platform. So I would encourage, if there's questions anytime, feel free to put them in the chat and I can I can answer them at the end, or I can stop and answer them. I think that, you know, as it comes to your mind, it's better to get the question down than to wait to the end. So what are, you know, just to emphasize what we're doing with nanovials, right? These are these hydrogel particles. They have about a 1 nanoliter volume inside of them. We create the suspension of these particles, you know, 1 million or so, in a vial or a tube. The particles themselves have functionality. They're a hydrogel that can be functionalized. We usually functionalize them so that they can capture, they might have antibodies to capture things that are secreted. They might also have ECM components, extracellular matrix, or peptides that allow for cell adhesion. We can bind and grow cells on them. We can then exchange, similar to, you know, centrifugation and solution exchange operations that you would do with cells. You can do that with these nanovials. So you can exchange reagents across them, and then you can seal them and unseal them using this emulsification process, where we use oil, mix the particles with the oil and water to form an emulsion, and you get what we call a dropicle, a particle in a droplet. And then you can break the emulsion to recapture the particles. We can then stain, you know, through these solution exchanging operations, stain what might be accumulated on the surface of the nanovial. So that, let's say, it has a fluorescent tag, and then analyze and sort the whole particles and the cells that are contained using fluorescence activated cell sorters to come with different populations of nanovials. So this is the generalized workflow for using these nanovials with flow cytometry. So how do we make nanovials? So this is the only part of the talk where I'll actually talk about using microfluidic devices. So we use microfluidics to make the nanovials, and how we do that is basically co-flowing two materials, a multi-arm PEG with photoinitiator. Often this PEG is functionalized with something like biotin, so that we have a handle to build chemistries off of, and as well as dextran and a dithiol crosslinker. So these co-flow, they form droplets with this PEG and dextran, then going through what's called aqueous two-phase separation. At certain concentrations of PEG and dextran, you'll form a PEG-rich phase and a dextran-rich phase that are phase separated. We can then, you know, as these droplets form in this phase separated form, we can apply UV light to initiate the photo cross-linking reaction. The photo cross-linking reaction then gives us, after washing steps, we get, and breaking of the emulsion, we get these particles that have this bowl-shaped structure. And we can get, you know, suspensions of these particles to use for our assays. So this suspension of particles, so this is done, you know, once, and you make millions and millions of these particles, and those millions of particles then are used by life science researchers to do the assays. So it's kind of, the microfluidics piece is separated out from the assay piece. We can make these particles very uniform using this process. So here you can see opening diameters and particle diameters that are, you know, with CVs less, 2% or less. We can make particles of many different sizes. Here I'm just showing one size, but and they're very uniformly labeled. This is showing streptavidin labeling of biotin coated particles, and you can see very uniform fluorescence intensity. As I mentioned, you can make particles of many different sizes, and normally we're making them across the size range from about 35 micron outer diameter to 105 or so micron outer diameter that are suited for different types of cells. So smaller particles more suited for smaller cells like B cells and T cells. In the intermediate sizes, we can use human embryonic kidney cells or CHO cells, macrophages, mesenchymal stem cells, neural stem cells, et cetera. And then the largest sizes, we can all grow up colonies and clusters of cells so that we can perform assays on single cell colonies. These are compatible with a number of different cytometry instruments that we've shown so far, including, you know, the BD instruments, Sony instruments, et cetera, as well as some of the larger particles are compatible with other large particle flow sorters that are commercially available, BioSort or on-chip sort. So the first step in performing an assay with these nanovial particles is we need to load a single cell assay is to load the cells onto the particles first. And so how do we do that? We have different techniques. Generally, we're loading nanovials into wells or into tubes. We then load the cells into those wells and tubes, and they randomly will be loaded into the nanovials. And I'll talk more about that, but we go through a Poisson loading process. But we want the cells to bind irreversibly, well, not irreversibly, we want them to bind in a way to the nanovial so that during processing steps, they don't come off. And so we have different approaches that we've used to do this, using peptides that bind to integrins like RGD, polyalysine or fibronectin or other ECM elements. We can biotinylate cells' surface proteins and directly link cells that way, or we can include antibodies on the nanovial to cell surface markers. And all of these have pros and cons and apply to different cell types. So this is showing some of the data for, as I go from the left to right here, the most adherent cells that we tested down to cells that are the least adherent, that are usually grown in suspension. So these CHO-DB12 cells, we used RGD and they adhere well. We can load these cells using RGD as the adhesive moiety. XP-CHO cells, we had to use some combinations of ECM to load them effectively. HEX cells, we showed that we could biotinylate them, and that would allow us to load them into the nanovials. And then B cells, we used antibodies to CD19, CD45, or a combination of CD19 and CD45 in order to load them effectively. So because of the unique shape of the nanovials, if we let them relax or settle into a well, what you see is that they settle face up. The cavity will settle due to gravity because of the non-uniform center of mass. The cavity will settle with its face up. So in essence, you're creating in a well, you'll create millions or hundreds of thousands to millions of little nano wells that you can then load cells into. And so we let one of the approaches that we've used initially was to let the particles, these nanovials, settle, and then we would seed cells on top, and we'd seed them at concentrations so that followed some expected value of loading, so that we could go, in this case, lambdas of expected values of 5% of nanovials having a single cell, all the way to 30% or so. Lambdas of 0.3, and so they would follow Poisson statistics quite nicely. So, if we wanted to avoid multiplets, we use usually lambdas of 0.1 or so, and we get about 10% of nanovials have single cells, and very few have doublets. And as we go to higher lambdas, then we get a higher fraction that will have 2 or more cells. We can surpass Poisson loading in some cases by using other nanovial formulations that we've developed in the lab. In this particular formulation, we localize gelatin just to the cavity region. And so the adhesive moiety for cells is only in the cavity region. And so we can use the cavity in this case as a steric filter for cells, only one cell, let's say, to bind. And then now more cells can't bind, for example, around outside of the cavity. And this is important for very adhesive cells. This is a really adhesive cell. And if we have a uniform coating, you can see that cells start to adhere on the outsides. However, if we use this localized coating, then we get cells sticking generally just inside of the cavity. So this is an advantage. And that allows us to then also use the cavity size itself to preclude additional cells from loading. And in this case, we can actually beat Poisson statistics. Let's say here's the fraction for these 55 micron particles of 2 cells or more is following Poisson statistics. And now we start to get lower and lower doublet formation, beating Poisson statistics as we basically narrow the cavity size and become a smaller cavity closer to the size of cells. After we load cells, We have this optional step where we can form droplets. We can basically surround them in oil so that there is a fluid volume in the cavity, but there's a little crosstalk across between nanovials. This process of forming compartments, we use an oil and surfactant, mix it with our nanovials, and we pipette for about a minute. And through that process, we form this emulsion where the emulsion droplet size is templated by the particle size. So here you can see what this emulsion looks like, and you can see this uniform droplet size that are templated around the particles, and those are our dropicals. Then you see a bunch of other little droplets, which we call satellite droplets, that are around. We actually aren't concerned with those droplets because there's no solid phase, and there's no and so when we do all of our centrifugation and washing steps, we basically aren't capturing anything in those droplets, and they get removed in the process. So this process of forming droplets around particles actually was pioneered in the Richard Matthews group in 2011. And so it was great that we could build upon their technology, although in their case, they didn't have this nanovial morphology, right? They were looking at basically spherical hydrogel particles. The process of forming droplets also creates relatively uniform droplets. So here you're seeing the nanovial in red, the encapsulated aqueous fluid volume in blue fluorescent dye, and the oil is in black here. So here's several dropicals. The CV, if we image and characterize the volume of these dropicals, the CV is about 4%, which is similar to the droplets that are formed using microfluidic devices. And we have very few of these dropicals that have more than one particle. You can see one here where you have 2 in one droplet, but this is usually less than 1% of the droplets that are formed. I'm going to go briefly through this, but it's interesting that, so why do we get such uniform droplets that form with uniform volumes? It turns out that the system, as you agitate it, will minimize energy. And it turns out that the minimum in energy for this the system of particle, droplet, and oil is somewhere where you have basically a droplet that forms almost a sphere around one particle. So if you look at the volume of fluid that a particle is holding and the system energy, it starts out actually higher energy. This is the configuration. If you had just a little bit of volume in the particle, this is actually higher energy, and it decreases as you go to this normalized volume of 1, which is about just a sphere here, and then it increases further as you build fluid on top of this, and the surface area increases. So if you compare this volume, what we call volume energy curve, to the volume energy curve for a spherical droplet, this will just follow this constantly increasing volume energy curve. So what that is important is that when you have this constantly increasing volume energy curve, that means that two droplets will always want to coalesce, right? And we know that that's the case that the emulsion is coarse in. But in the case of these types of volume energy curves, where you have these changes in inflection of the volume energy curve, what we see is that if you have 2 droplets that were splitting, that you get actually interesting dynamics where you get one droplet that takes all of the volume, and then you get the other droplet that will basically peg out at this preferred volume near a normalized volume of 1, or asymptotically approaching the normalized volume of 1. Now you can imagine if this droplet splits again, it can go through this process over and over as you're agitating and add more energy into the system. And so over time, you get more and more of the dropicals accumulate at this volume of 1. And so this is a kind of the mechanism that we see for why we form uniform droplets around these types of particles. Okay, so what kind of assays can we perform with these nanovials? So let's go into it. So first, I wanted to show you the types of the analysis approaches that we use with flow cytometry. So here we have, just to give you some reference, we had free cells that were in this pink color, magenta color. Empty nanovials, those are in this green color, and the nanovials with cells, and they were labeled separately so that we could identify them. So in this experiment, you can see, in the fluorescence channels, you can identify these subpopulations. And so how do they look in forward scatter and side scatter channels with fluorescence, with a flow cytometer? And you can start to pick out, actually, the cell-loaded nanovials have both high side scatter, which is associated with these empty nanovials, as well as higher forward scatter associated with cells. So you could start to pick out this population, and you could sort them and get a pure population of cell-containing nanovials. Okay, so where are we applying this? I mentioned monoclonal antibodies, and so we have some applications both in production, developing the cell lines that produce monoclonal antibodies, and how do you select cells that produce high levels of a therapeutic, as well as in the discovery of monoclonal antibodies. How do you find, let's say, B cells or hybridomas that are producing antigen-specific monoclonals that are going to be potential drug for potential drugs, and then you could sort those out, sequence the heavy and light chain of the antibody so that you could produce those in mass as a drug. And so this is relevant, I think, for all of us now as we have been developing monoclonal antibodies to treat SARS-CoV-2, and how rapidly that happened. And actually, that there are some of the initial drugs that were approved or authorized in the U.S. were developed using microfluidic approaches, where they took B cells from a patient, they identified the antibody secreted from those particular single B cells that bound spike protein and potentially blocked its interaction with ACE2, and then we're able to sort out those B cells, sequence them, and then develop scale the production of those monoclonals for use. So that's one area that I'll show some example application. The other is in cell therapies, where we're interested in sorting out T cells that are secreting cytokines that suggest that they're engaged and active. And so this can also be used in discovery of new CARs, discovery of new TCRs, as well as monitoring of cell therapies for function. And then finally, just some brief snippets of what we're doing with characterizing cells based on the production of extracellular vesicles and growth factors like VEGF. So how does these assays work? This is kind of a generalized workflow for identifying the production rate of IgG from single cells. We first load, in this case, production cells, CHO cells, into the nanovials using RGD. We add an anti-IgG capture antibody on the surface of the nanovial. We encapsulated in oil, allowed the cells to secrete, accumulated the IgG that was secreted by that cell, broke the emulsion, added a fluorescent antibody that bound to the secreted IgG, and then we could sort out those populations of high secretors. So this is kind of what you would see. You have some antibody producing CHO cells where you get high green fluorescent signal around where the cells are, and then some non-producing CHO cells that were in the spiked solution of cells and that are in blue, and then you see no antibody signal. And then after you do a sort and enrich, you can see antibody signal associated with the cells that were spiked in that produce antibody. So what we did for this assay is we wanted to look at, okay, if we could sort out these populations of high antibody producing cells, would they maintain that production capability? And so could this be used then to improve the capacity to manufacture monoclonal antibodies for therapeutics? So you start with your initial assay. You have cells that are seeded into the nanovials. You perform the IgG staining. We then run through the flow cytometry system. We look at this top right quadrant, which is cell positive, as well as anti-IgG secretion signal. If you take this top right quadrant and we look at it as a histogram, we then looked at taking all of the secretors, all of the IgG positive nanovials, as well as the top 20% of secretors. We could sort out those populations. This is all the secretors. You can see, okay, yes, there are some that secrete more than others, or if you take the top 20%, you can see that the signals were generally higher. We then expanded these populations over a week and then looked at the bulk production of IgG for the expanded populations. And we compared the pre-sort populations of CHO cells to all the secretors and to the top 20%. And what you can see is that, yes, what we found is that this secretion phenotype that we were selecting based off of, that it was maintained at least over this week period. And we could get up to a 50% increase in production from around 40 to around 60 nanograms per milliliter in this particular assay and time period. And so that's a huge increase if you think about it. If your manufacturing capability is now increased by 50%, the efficiencies and the costs go down dramatically for producing these monoclonal antibodies. So we've done these assays not just using the droplet encapsulation approach. Remember, I talked about the gelatin nanovials that have the localized signal within the cavity, the localized capture and adhesion within the cavity using the local gelatin. So what we found is that using that approach, that we didn't need a droplet encapsulation step because we had less crosstalk. And so in this format, we have capture antibody that's added. Cells are bound, capture antibody is added. We then incubate the cells, allow them to secrete without any drop formation. And then we wash and add fluorescent IgG and then sort out populations. We could sort about 100,000 cells easily in an experiment based on secretion using this approach. And what you see is that within the first hour or two, you actually don't get much crosstalk. That means that the cells that are secreting, that they don't crosstalk to the neighboring nanovials. They accumulate mostly on the nanovial in which they are located in. And you see this here. So we labeled empty nanovials and cell-loaded nanovials. What you see is that at 0.5 and 1 hour, the majority of the cell-loaded nanovials have signal above the top 99% of the empty nanovials. Over 4 hours, then you can see the empty nanovials, the fluorescence intensity starts to shift up as you get some antibodies that are diffusing out and binding to neighboring nanovials. But this is great that we could then sort, even after 1 hour, we could sort the top 5% and get enriched populations of IgG secreting cells. One thing that we found is that when we load cells in these nanovials, that it prevents some of the damage that occurs during FACS sorting. So if you take CHO cells and you put them into suspension and you sort them using FACS, and these are droplet and air sorters, you have viabilities that are approaching about 60% or so for the free cells. They get damaged during the sort process. If the cells within a nanovial, we could improve the viability post-sort to 80% or so. So we had this improved viability, potentially because cells, first, they maintain their adherent state during the sorting process. And two, shear stresses during the sorting process are mitigated by the cavity. The cells are kind of protected because they're in a cavity. We also found that during the flow cytometry process, you can get improved signal to noise on the nanovials compared to microscopy. And this gives you, this is another advantage for using equipment that's already in the lab that's extremely sensitive. Flow cytometers use photomultiplier tubes to do fluorescence detection compared to microscopes, which are usually using cooled CMOS cameras. And so we can get a linear range over several order of magnitude and detection limits down to about 10,000 molecules using flow cytometry to detect the molecules attached to nanovials. We can sort up to 1,000 nanovials per second as well. And this is an improvement over using microscopic techniques to do the analysis. Okay, so I showed you the assay format for characterizing the antibodies secreted by a cell and how much IgG that cell produced. In this assay, we modified the format a bit, and instead of characterizing just the amount of antibodies secreted, we also wanted to characterize whether that antibody was specific to a particular antigen. And so in this case, we add a fluorescent antigen in the final step to label the secreted antibodies. And then we could sort out the populations of cells that secrete antibodies that are specific to that particular antigen. And so this is the discovery approach, right? So this is not, I'm producing monoclonal antibodies. I already know the sequence. I already know what I want to produce, and I just want to get a cell line that produces it more. The application here is I want to find out what antibody sequences give me antibodies that will bind my antigen the best. And so the format, very similar, as I mentioned, we capture B cells using CD45 antibodies. We have anti-IgG on the nanovial also to capture the secreted antibodies, and we add fluorescent antigen and then sort to do single cell assays downstream, like single cell RT-PCR to capture the heavy and light chain information. We started off by using hybridomas that secreted an antibody to hen egg white lysozyme, and so HEL. And so this is the format, so secreted IgG, and they had fluorescent HEL. And then, we performed this. We, started off with just, kind of a pure population. And yes, you could see that, the hybridomas that bound gave you this nice fluorescent signal where they bound, and we could sort out these populations and enrich them. We next then performed basically a spiking assay where we had the HEL hybridoma spiked into a background of another hybridoma line that did not bind the HEL. And so this was like our, think about it as kind of simulating if you're finding a rare population of antibody secreting cell that secretes against the antigen. So once we did the spiking experiment, so this is kind of the pre-sort, you can see a bunch of 90-10 hybridomas and a few of these high HEL5 that secrete the HEL, anti-HEL antibody. We would gate out the nanovials, we gate out the top 0.18% of HEL 647 positive nanovials. And that would give us these populations of cells that get sorted. And what we found is that we could enrich from a 4% or so initial pre-sort concentration of HEL to up to 90% plus post-sort concentration. So this is a great enrichment of the antigen-specific hybridomas using this process. We could then do downstream processes with these sorted cells. So we could do RT-PCR to capture, in this case, I'm showing the heavy chain amplification, or we could perform clonal expansion. So this hybridoma grows out over 3 days to kind of cover the nanovial and then continue growing. Now you can see a few more of these like clonal populations growing up after sorting. So here you can kind of see them grow off and basically start to fall off the nanovials, and it'll establish a new colony. So this is a good way that you can also amplify your signal for capturing sequence information. After we did this assay with hybridomas, we then looked at identifying B cells from mice immunized with an antigen. And so in this case, we used ovalbumin as our antigen. We then immunized mice with ovalbumin. We would isolate after 3 weeks B cells from the spleen, and then we will characterize and isolate antigen-specific B cells. We also isolated serum, and just to make sure that the cells were that had produced antibodies against ovalbumin, we would test that on our nanovials, and what we found is yes, that the serums were positive for anti-ova antibodies, and so we went ahead with the splenocyte characterization. So we could load the cells from the immunized mice onto the nanovials. We could then sort out the top 2% in this case of nanovials based on fluorescence, and we saw a few different types of signals. We would see secretion signals, so the nanovial itself becomes very bright. We also see some cases that cells were bright. In some cases, we have B cells that have a B cell receptor that's binding antigen, and we could isolate those populations as well. So we could isolate cells that were secreting highly, plasma cells or plasmablasts, as well as other cells that had antigen-specific signal on the surface of the cell. And so that gives us a more diverse population of B cells that we could then isolate and get and understand what the sequence information was that made them antigen specific. So the other assay, so moving on from B cells, so that this shows that the proof of concept that we can make a workflow that allows us to isolate antigen specific B cells and then get sequence information from those. You can imagine that the nanovials, there's many different formats for those types of workflows. And so that's something that we're pursuing for drug discovery applications. Also for T cells, we can do a similar type of secretion profiling. In this case, not based on the antibodies that they're secreting, but based on cytokines. Again, we use CD45 to capture T cells. We have a cytokine capture antibody on the nanovial. We capture cytokine at a secondary that binds and sandwiches the cytokine. And then we have fluorescent signal that we can sort out T cells based on cytokine secretion. We did a number of different cytokine assays that we developed: IL-2, TNF-alpha, interferon gamma. I won't go into details here, but all of them, we get down to 100 nanograms or less sensitivity. And then we performed assays on T cells. So here I'm showing TNF-alpha and interferon gamma. You can see, here's a T cell in this nanovial, and you can see the fluorescent signal associated with the secretion of interferon gamma. Or here's another T cell and a fluorescent secretion associated with TNF-alpha. We could sort out these populations of high TNF-alpha or high interferon gamma, and then we can enrich them post-sort. So one thing that's interesting here, TNF-alpha, there's two forms of it. There's a form that's bound to the surface of a cell, and then there's a secreted form. And we could actually see both of these with the assay. So here you can kind of see the cell surface is not brightly lit, but you see some secreted signal. Here you see a surface signal as well as a secreted signal, and here is only a surface signal. So this allows us to look at different populations and really to try to classify these cell populations in ways that haven't been possible before, because you can characterize both secretion and surface-bound forms. How we can characterize those differences is actually using some tricks with flow cytometry. So it turns out when a cell or a particle, a nanovial, goes through the laser of a flow cytometer, you don't just get one intensity value, right? You get a peak of intensity, and that peak has both an area or a width associated with it as the thing passes through the peak at a certain velocity, and a height. And so what we do is we use some peak analysis to characterize whether that peak was associated with signal on the nanovial itself. And those peaks that have signal on the nanovial themselves, they had a high area for a given height. So here on the y-axis is the fluorescent area, and this is the fluorescent height. So nanovial specific peaks, they're wide and have a large area. And so we can sort those populations out that have nanovial specific signals using that area. And then the peaks that were high in height but low in area, they associated with punctate signals on cells. And usually this means like dead cells. And so we could sort those out separately as well. But mostly we're interested in this population. Okay, so this shows the kind of proof of concept for characterizing activated T cells based on their secretion of interferon gamma, TNF-alpha. And so this is an exciting direction if we want to move into functionally defining populations of cells for cell therapies, rather than saying, oh, it has this surface marker. We want to know, what does it do? Is it secreting this? Is it killing these types of cells? And nanovial approaches, lab on a particle approaches allow us to do that. We've been moving this assay, in the last few minutes here. I'll kind of just rapidly go through a few different types of assays. One that we're doing is, okay, how do we characterize cells that produce other types of secretions like extracellular vesicles? there's very little that's understood about what makes a cell produce more extracellular vesicles. What's the biogenesis of these? Can we also develop cells that are high producers of EVs as, lines to manufacture extracellular vesicles as therapeutics? And can we characterize, the heterogeneity in EV secretion as a biological effect? So these are things that can't be done now because we don't have ways to associate the EVs produced by a same single cell. So we've been developing technologies using our nanovials to do this. We capture EVs using CD63, for example. We then label with CD9. So this allows us to characterize CD63, CD9 double positive EVs secreted by these particular cells. We perform the assay. We then add antibodies, CD9 antibodies as fluorescent so that we can sort out these populations. So just briefly, we can pull out these populations of EV secreting cells, and we can sort them out and you can see, the EV signal here associated with cells, for example, as well as like EV specific signal here, in the cavities. One thing that we have to do is, because some of these markers also will stain cells, we use the height versus area metrics, as I mentioned before, to pull out these subpopulations that are associated with high EV secretion and not just the staining of the cells. And also VEGF. So we've been using mesenchymal stem cells that secrete vascular endothelial growth factor. And so what we are able to do here is, similarly to the cytokine assays, we form a sandwich, and that allows us to secrete, to sort out subpopulations of these stem cells that produce high levels of VEGF. And so that we're regrowing these populations of stem cells and try to functionally define a population of MSCs that might be better for regenerative medicine because they produce high levels of this growth factor versus just having a random population of cells being used in these regenerative medicine applications. Okay, so I think I'll try to wrap up here. What we're truly trying to do here with these nanovial platforms is, again, this is the technology is something that any research lab can use. It's not require a specialized instrument, just the flow cytometers and fax instruments that are already present in lab, centrifuges, pipettes, things like that. There's no microfluidics. That's all done at one time to make these particles. And so we think that this can help democratize the ability for research to occur in single cells, especially on the functions of single cells. And I showed you mostly functions associated with secretions, which actually encompass a huge number of applications in what I call these 3 pillars of medicine, in molecular therapies, gene therapies, and cell therapies. Microfluidic technologies have tried to address these issues, but there's been challenges both with throughput and the accessibility and the ability to sort. And so we think that droppicals or these nanovial approaches can, certainly have higher throughput. The throughputs are limited by the throughputs of flow cytometers, and they have these capabilities that these other technologies didn't have in terms of, for example, characterizing adherent cells like mesenchymal stem cells, which I just showed you. So this is super, we're very excited by the technology, and we've been moving it in many different directions. And, or if anyone is interested in using these technologies, happy to talk to them. And with that, I'd really like to thank you for your time, and also thank my students, in particular, Joe Derete, who was one of the students who did a lot of the work in developing the nanovial technology in the lab. Also is now the president of Partillion Bioscience. If you're interested, Partillion is hiring, in particular, we're actually hiring microfluidics engineers at this time as well. So with that, I thank you again for your time, and happy to take some questions.
Thank you, Professor Di Carlo. That was an interesting talk and a very exciting technology as well. Let's hear some questions from the participants. Before that, I have a question. how do we control the size of these nanovials? You talked about nanovials of different sizes,
Right.
small, medium, and large. What controls the size of the nanovials and the cavity as well?
Yeah. So those are good questions. So the size of the nanovial is controlled by the droplet size when we use the microfluidic devices. So that we can control by the flow rate ratios of the oil to the aqueous phases or the channel sizes. So we'll have slightly different microfluidic devices for the different sizes. But the cavity size is controlled actually by the ratio of the dextran to the PEG phase. So there we have to tune the fraction of dextran versus the PEG that's included in the final droplet. And if there's more dextran, that will lead to a larger cavity. If there's less dextran, that leads to a smaller cavity.
Okay. And you're talking about dropicals. What about the stability of those dropicals? How stable are they when it's an emulsion? So how stable are they?
Yeah, so when we form the emulsion, we're also adding surfactant. So we do add surfactant to prevent emulsion coalescence. And that's for these nanovial emulsions. Actually, for other dropical emulsions where we have a hydrophobic outer region and then a hydrophilic inner region, we actually don't have to add surfactants, and they're stable in that situation too, because of the unique structure of the particle. But yeah, so the surfactants help in the case of the nanovials, which are hydrophilic hydrogels. And then for the other particles, these kind of concentric particles that have PEG and polypropylene glycol, they are stable without surfactants.
Okay, thank you. So questions from participants?
Yeah, so there seems to be a question. So how can you remove the cells from the nanovial?
Right.
So I showed, so this is a good question. So I showed some data where as the cells grow, they kind of grow off and fall off the nanovials. That's the preferred way that we remove them. We just let them grow off. But you can also trypsinize the nanovials. So think about it as like a surface of a well plate, and particularly like the gelatin nanovials, they have a gelatin layer. You add trypsin, it cleaves the gelatin, and then the cells come off. Yeah, I think actually one great way to think about the technology is a well plate for a single cell. So it's like a floating well for a single cell.
Okay. So let's wait for some questions. Yeah, there's another question.
So the question is about the monoclonal antibodies. Can you separate them? So I guess the question would be whether you could use this as like a filtration technology. I'm not sure exactly, but so I think the main use is in characterization of the amount of secretion of a cell, and then sorting that cell so that you can then now have a population of cells that secretes a lot. The application of like then isolating the antibodies that are secreted, I think there are filtration, like chromatography columns and things that are used to purify antibodies from like cultures. So those might be, I don't know if we can use the technology in the downstream production phase, but definitely in the phase of selection of your cell line for production.
Okay. Yeah. Okay.
But it's interesting, right, that my presentation today, very little microfluidic, traditional microfluidics. Hopefully that sparks the imagination a bit.
That's right. Anybody else has questions on the participants? Yeah, participant from UCL, UK. Questions? Any questions from Priyankan? Yeah, just question from me, Professor Di Carlo. You had shown a schematic image on a settling behavior. I didn't get that image clearly.
I can try to pull it back up. Hold on.
Yeah, sure.
This is an image.
Yes, this is a slide, yes.
Yeah. So it's quite a nice effect that they will settle and then they reorient their faces up. So that helps with the loading process, because you can imagine the loading is not, you'll have some loss of cells if there's many face down nanovials. So this loading approach of settling and then cell loading is what we initially used, but actually now we do even simpler approach. In a tube, we just mix cells with the slurry of nanovials and then let them settle in a kind of three-dimensional pack. So not even in a monolayer, and we get actually high loading fraction in that case too. So even simpler than this process, but this is what we initially were doing.
Okay. I think you have a question from the okay. Thanks. Okay. Okay, it looks like there are no more questions from the audience participants. Professor Sarath, any questions?
Thank you so much. Appreciate your time today, and hopefully there was. Was there more questions? Sorry.
I don't think so. There are any questions?
Okay.
Yeah. So let's thank Professor Di Carlo of taking time. It's Sunday late evening for him. So really thanks to take off your time and give a nice presentation. And thanks from all of us here.
Thank you, Professor Ranganathan, for the invitation. And hopefully someday I can see you all in person. But it was nice to meet you virtually today.
Certainly, we'll take an opportunity to meet you. This question looks like, can we also control the orientation of cell within nanovials?
That's a good question. So what we can do in the nanovial is make an adhesive surface. So let's say that there is a cell that has an apical and basal side, then the cell will start to form connections, let's say extracellular matrix proteins or peptides on the nanovial, and then it will orient. It will have like a apical and basal side to it. For other cells, that are generally in suspension, B cells and T cells, they don't really have an orientation, right? So then they will adhere in kind of a random configuration. But yeah, I'm not sure if that answers your question, or if you meant more of like where on the nanovial cells are.
Okay.
Great.
Okay. Thank you again, Professor Di Carlo. See you
Thank
sometime.
you. Have a great rest of your week. Take care.
Yeah. Bye.
Bye-bye.
Yeah. Thanks everybody for participating. Thanks all the participants.